I'm going to get a little deeper into the DAT now. In our last video, we talked about IAT versus the DAT and when we would do both and the differences between them. Now I'm going to get a little deeper into the DAT. So if you remember from the last video, the DAT, we are looking at what is happening in vivo. We're looking for those cells, the antigens, and the antibodies to attach to each other, become sensitized in the person's body. So we draw the blood. In our picture here, I'm showing an example of a transfusion reaction. The red cells are the patient's original cells. The blue cells are going to be your transfused cells. So what happens is those blue cells, the patient's antibodies attach to those blue cells, and we draw the blood, and the tube of blood has some of both. So what we're going to do is do the test, and we're looking for little microagglutinins of those donor cells. So let's talk about how we do these tests and what we do. First off, we're going to talk about the polyspecific tests. The polyspecific tests, we are going to use your AHG / anti-human globulin / Coombs sera / the green stuff, right? The stuff that's in your rack that we use all the time for antibody screening and any of our HG cross matches, things like that. So we're going to use that. That is called polyclonal AHG. We have anti-IgG and anti-complement both in this vial, in this antisera. So what we do is we make a wash cell of the patient's cells. We take 2 drops, we put it into the tube, and we wash it one more time. When we get that out of the centrifuge, we're going to decant it and we're going to blot it really, really good. So don't forget, we always want to get all that extra saline off of that lip prior to adding the AHG, because if I don't blot that well and I turn it upside down too soon and there's too much saline in the bottom of my tube, the AHG becomes diluted and we could have a false negative or a weaker reaction than expected. So we put the 2 cells in, we blot it really well, and then we're going to add the AHG to both tubes. You're going to mix it, because these cells, there's going to be very few cells on the bottom, so you're going to mix it. The poly 5, just leave in your rack, because we'll come back to that in 5 minutes after the incubation. The immediate spin, though, we're going to mix it, we're going to centrifuge it. Then you're going to look at it under your agglutination viewer. Remember, we always read everything under our agglutination viewers. It is an FDA requirement. So we're going to look at that under our agglutination viewer. If it's negative, we're going to take 1 drop. Use your plastic pipette, put a drop on the slide, put a cover slip on it, and we're going to take it over to the microscopes. We're going to look at it under the microscope. We are looking for these tiny little agglutinins among a sea of donors, of patient cells, because the donor cells will all agglutinate if they have the antibodies attached in vivo, and the regular patient cells will all be loose cells. If it's negative, we're going to go ahead and add our Coombs check cells, and it better be positive at this point. If it is still negative, something went wrong in the test and you need to repeat your test. By then, your 5-minute incubation is over. Give it a little mix, centrifuge it, run it through those same steps. Read it under the agglutination viewer, put it under the microscope if it's negative. If it's still negative under the microscope, add your Coombs check cells. If both sets are negative, if your immediate spin and your 5-minute are both negative, you're good to go. Your DAT is negative, you can stop. If, however, either one of them is positive, now we have to go to monospecific testing. Monospecific testing, what we are looking for is the individual IgG or complement. So we break those up. So remember, this is both IgG and complement in the same tube. Down here, we're going to make a new wash suspension. The reason we have to make a new wash solution is over time in that saline, those antibodies or complement can be eluting off the cells. And we want to make sure that we do not miss something that's there. So we go into the original tube, make a new 3%. We label 3 tubes, IgG, C3, and saline. Now, don't forget, on my board here, I do not have the patient information. You will have patient information on every single tube that you do. Do not forget. So I have my IgG tube, my C3 tube, my saline. I take 2 drops out of my new wash suspension. I'm going to put them in all 3. I'm going to do 1 more wash, and then I'm going to decant it and blot the tube. Into my IgG tube, I'm going to put the anti-IgG. Into my complement tube, the anti-C3. And then in my saline tube, I'm going to drop 2 drops of saline, just normal saline from your wash bottles. Give them all a nice mix. Immediate spin, saline, and the IgG, we're going to do immediate spins for those. We're going to spin it, look at it macroscopically with the agglutination viewer. If it's negative, look at it microscopically in the microscope. If it is still negative, you can use your regular check cells. Don't forget, our regular check cells are IgG coated check cells. Those are the ones we always have in our racks at all times. So we can use those for the polyspecific and for the monospecific IgG. Saline does not get check cells. We only use check cells when we use AHG or anti-IgG, anti-complement. We don't do check cells otherwise. And remember, the purpose of the check cells is to make sure that the AHG was working, to make sure that you remember to add the AHG. It's just to make sure everything's right. If your check cells do not work, the whole test is invalid and you have to start over. Because if your check cells don't work, how do we know that your patient was truly negative? Maybe they were positive and something was wrong with your AHG. So, immediate spin IgG, your regular check cells. Immediate spin saline, no check cells. After your 5 minutes, you're going to incubate this one. Then you're going to spin it. You're going to read it macroscopically. If it's negative, microscopically. If it's still negative, you're going to make sure to use your complement coated check cells. Those are a special check cell that is in the DAT rack that we only do for anti-complement monospecific testing. They get their own special check cells. Assumedly, if the polyspecific is positive, one of these should be positive. If not, I suggest starting and repeating everything again because something went wrong. We only go on to monospecific testing if the polyspecific is positive, and it would only be positive for either IgG or the complement up in the AHG. So make sure, a couple take-home points. Make your new 3% wash suspension. When you spin those 2 drops, blot them really, really dry. Make sure to use your correct check cells for whichever tests you're doing. If you have any questions, be sure to reach out. I'm happy to answer anything I can.